Okay, today I'm going to talk about a very important concept by the name of DNA supercoiling. And you know that this topic is really important because it is somewhat related to some processes like replication and also recombination. So that is why it is important to know that. First of all, we should know the basic structure of DNA. That, as you can see, that it is a double helix form structure. That it is made of backbone, right? That it is made of phosphate and sugar, but between them nitrogen spaces are present. That the guanine with the cytosine and adenine with the thymine, they have a connection with each other by hydrogen bonds. And every of them it is called one base pair. One base pair, one base pair, and like this you can count them. In the structure of DNA, this is called a minor groove, but this groove is called major groove. That this is also important to know that this is major groove. You can see from a structure, right? And this is minor groove. The important concept also is relaxed DNA, because before knowing the supercoiling, we should know that what relaxed DNA is. Any DNA which exists in most stable state without any bending on itself or upon itself or upon any other protein, so that is called relaxed DNA. For example, you can see that this DNA is in circular form without any bending on anything. So this DNA is completely relaxed. Or we have another definition that if in one turn of double helix of DNA just 10.5 base pair are present, so that DNA is called relaxed DNA. And in this DNA, the minor groove, from one minor groove up to the other minor groove, or from one major groove up to the other major groove, every of them it is called one turn. That if the number of base pair are 10.5, yes, that is called relaxed DNA, or that DNA is completely relaxed without any supercoiling. And because the most common structure of DNA, which is the most common structure of DNA is the beta form structure that Watson and Crick said. So that is called relaxed DNA, because it is the most common form of structure of DNA. We have a quiz. In your idea, this DNA is relaxed or not? Yes, you can pause the video and then answer. Yes, this is relaxed DNA. But what about this DNA, and what about this? Yes, this DNA is completely relaxed without any bending upon itself or in any other protein. But this DNA is not relaxed DNA. Why? Because in this point, and also in this point, it is supercoiled upon itself. So this is not relaxed DNA. The other important point that we should know is twisting of DNA. Counting the number of times the two strands cross each other completely, that is called the number of twists which is present in that DNA. For example, in this DNA, you can calculate the number of twists that if we count all the number of base pairs which are present in this DNA, for example, 21 base pair, and then divided them to 10, and then we divide them by 10.5, so it will be equal to 2. It means that the number of twisting is 2. But the question is this, that this DNA is twisted in this direction, or no, in this direction. Okay, that is why we should know the types of twisting. According to the law of handedness in physics, you can know that if we place our right hand in this confirmation that our right thumb is pointing up, and then by our right thumb we follow this strand which is passing on the other strand. You can see that this is passing on other strand, upon it is present upon other strand. So, and we follow this from this groove, so our right thumb is pointing toward the left, so this is left-handed DNA. But this strand which is passing on the other strand, if we follow this by our right thumb, it is pointing toward to the right-handed direction, so this is right-handed DNA. And also you can know it from this word that clockwise and anti-clockwise, that if a DNA is twisted according to the clock turning, so that is called right-handed DNA. But if it is twisted in a left-handed fashion, actually it is twisted anti-clockwise. It means the opposite of turning out the clock. And as you can see in this picture, that this DNA is twisted in a right-handed fashion, so this is right-handed helix. And but this DNA actually it is twisted in a left-handed fashion, so that is left-handed helix. It means that anti-clockwise, it is anti-clockwise turning, right? And because the right-handed helix we show them by positive sign and left-handed sign by negative, that is why that the twisting number it can be positive or it can be negative, according that DNA is left-handed or right-handed. And these words overwinding and underwinding, we will see lots of these words in different kind of book. That's why that we should clear our concept about them. The number of base pair per ternus, if that is less than 10.5 base pair, that is called underwinding of DNA. But if the number of base pair per turn is more than 10.5, that is overwinding of DNA. Okay, we have a quiz. In your idea, this DNA is right-handed, or this is right-handed or left-handed? As you can see that this strand is actually our right thumb is pointing toward to the right direction, so this is right-handed double helix. But this DNA is actually our right thumb is pointing toward to the left, so this is left-handed double helix. And what about this? In your idea, right-handed or left-handed DNA? Yes, as you can see that this is pointing toward to this direction actually, so this is right-handed DNA. Now we reach to the concept of supercoiling. Normally, as you can see, that this DNA structure is double helix and it is normally coiled. But if DNA is further coiled upon itself or upon any other protein, that DNA is not relaxed DNA, that is supercoiled DNA. As you can see in this picture, that it is further coiled in this point and also in this point. So this DNA is not relaxed anymore. So this is supercoiled DNA. And in this example, actually, this is relaxed DNA, but it is further coiled upon itself in this point, and also this. So this is supercoiled DNA. And the very important matter that always you should remember is this, that supercoiling occurs in closed DNA. That it can be kind of circular, closed circular DNA, or a linear DNA, which is wrapping around histone proteins or about around any other protein. So the supercoiling also can occur in that kind of DNA also. The type of supercoiling that this concept will help us to know that if this DNA is positively supercoiled or negatively supercoiled. That is why that most of the students they have kind of confused about this concept, but I try to learn you very easily. In inter-1 or polyctonomic type, and also in toroid in a spiral form or a spiral form, actually we have positive and negative supercoiling in every of them. First of all, what I mean by inter-1 or polyctonomic, if DNA is supercoiled around itself without any involvement of any protein, so that supercoiling is called inter-1 or polyctonomic. As you can see in this cord of telephone, that if you imagine that this DNA is circular DNA and it is supercoiled in this point, and also in this point, it means upon itself. So this DNA is inter-1 or polyctonomic. But in the toroid or spiral form, which the long axis is one in a cylindrical manner, that often it occurs when DNA wraps around the proteins. For example, in eukaryotic cell, the DNA is wrapping around the histone protein, right? Yes, and this DNA is toroid or spiral form, and you can see that this structure is like in a cylindrical manner. This one in a cylindrical manner, as often occurs when DNA wraps around protein. Okay. In inter-1 or a polyctonomic, we have a positive and negatively supercoiling concept. That in inter-1 or polyctonomic, as you can see this picture, that this is inter-1 or a polyctonomic type. Why? Because it is supercoiled upon itself, right? So this is inter-1 or polyctonomic. And then, as you can see, that this is further coiled in a right-handed fashion, so it will produce negative supercoiling. But if this DNA is further coiled in a left-handed fashion, so it will produce positive supercoiling. But in a spiral form of supercoiling, it is the opposite of inter-1 or polyctonomic. As you can see in this picture, that actually this DNA is further coiled, actually it is further coiled around this histone protein in a left-handed or anti-clockwise direction, so it will produce negative supercoiling. But if it was further coiled around this protein in a right-handed fashion, so it will produce positive supercoiling. So it means that this positive and negative supercoiling is in opposite form in inter-1 or polyctonomic as compared to a spiral form of supercoiling. Okay, this DNA is also, you can see it, right? That this is relaxed DNA, but this part of DNA has been changed to a left-handed toroidal, right? And this part of DNA is also supercoiled in a left-handed inter-1 right. Yes. And according to the explanation that I said in previous slides, so a left-handed toroidal right will produce negative supercoiling, but left-handed inter-1 right will produce positive supercoiling. That because just we are wrapping this DNA in a left-handed fashion around this protein, it will produce negative supercoiling. But for compensating this part of DNA will produce positive supercoiling, and you know that this is left-handed inter-1 right, that it can produce positive supercoiling, right? Okay, we have quiz. In your idea, this is positively supercoiled or negatively, and from which type it is. You can answer. It is easy. And, what about this example? Is it supercoiled or not? And what about this? Yes, this DNA is not supercoiled, it is completely relaxed. But this DNA, you can see it, that this is further coiled upon itself. It means that is interwound, but it is in further coiled in a right-handed fashion, so it will produce negative supercoiling. But this, but not in a spiral form. In a spiral form, it means it will produce the opposite of this type of supercoiling. It means that this concept always you should remember, because it is really important, and most of the students just they confuse about this concept, that you always remember that in interwound type, if left-handed further coiling occurs, so it will produce positive supercoiling. But if right-handed supercoiling occurs in interwound type, so it will produce negative supercoiling. And then in a spiral form, you know that this is the opposite of interwound type. The linking number, it is also important, because you should know it, that this concept is somewhat related to replication and also recombination, right? The linking number, it means the sum of twist and right. This it will produce a linking number. And always remember that linking number in a relaxed DNA, it is completely equal to the twisting number. It means that the writing number is 0, because we said that in a relaxed DNA, that DNA is not supercoiled. It is in most stable state without any bending or supercoiling. That is why the writing number is 0. In this example, as you can see, that this is the linking number is 1 and linking number is 6. Because these both of DNA are they are relaxed, right? Relaxed DNA, so the linking number is equal to the twist number. It means just this strand is crosses the other strand just in one point, so this is the number of twists is 1, so the linking number is 1, but the writing number is 0, because this DNA is relaxed. And also this DNA is relaxed, yes, that in 1.2, 3, 4, 5, and 6. It means in these 6 points, one strand crosses the other strand completely. That it means that the linking number is equal to the twisting number, that it is equal to 6. Never forget this concept that linking number of DNA does not change, because the twisting and writing number are interconvertible. What do I mean by this exactly? It means that if the number of twisting is increasing, the number of writing should be decreased. Or if the writing number is increasing, the twisting number should be decreased. Because the linking number should be stable, and there should not be any change in linking number. In every situation, but we have an exception, that if DNA strands are cut or broken and rejoining, that the most important example is topoisomerase enzyme. So in that time, the linking number will change and it will not be stable anymore. That you know that these topoisomerase, they can produce positive or negatively supercoiled DNA. So that in that situation, the linking number will not be stable. It means that it will change. As you can see in this example, that this DNA is supercoiled, but this DNA was completely relaxed. That you can see that the number of base pair are 360, and the linking number is completely equal to the twisting number, and writing number is 0. Why? Because this DNA is not supercoiled DNA. This DNA is relaxed. That is why the writing number is 0. And the twisting number, you can find it that you have the number of base pair in this circular DNA, and then you divide them by 10 or 11, and you can find that the number of twisting is 36 in this relaxed circular DNA. But when topoisomerase work on this DNA, so and in that situation, this DNA is, you can see that this DNA is supercut. And as you can see that this DNA is interbond type and supercoiling, and actually it is in a right-handed fashion, so it will produce negative supercoiling. That is why that the writing number is shown by a negative value. And how we can calculate the 4 number, you can see that in this point, in this point, in this, and also in this, it is further coiled. It means in 4 point it is supercoiled negatively. That is why minus 4, the writing number is equal to minus 4. And twisting number is it is 36, but the linking number, you can see that the linking number has been changed. It means that 36 minus 4 will produce 32. We said that in just this is exception that topoisomerase will change the linking number, but in every other situation the linking number is constant and stable, and it will not change. And also this DNA is, you know that this is relaxed, right? But if we wrap this DNA around this protein in a left-handed fashion, so left-handed toroidal, right, it will produce negative supercoiling. But in other in the lower part of this DNA, the positive supercoiling will produce. Why? Because exactly we said that the linking number, it should not be changed. So it means that this negative value, it will be neutralized the positive value. So the number of linking number overall, it is unchanged. And it is true that there will be some changes in linking number, but overall the linking number will be unchanged. Okay. The other concept is linking difference. That the linking difference, by this concept we can know that a DNA is positively supercoiled or negatively. That just we should find the linking number in the strand, or in the, we can say in DNA that is positively or negatively supercoiled, and then minus the minus that the linking number, it means which is present in the relaxed form of that DNA, and then it will produce linking difference, that it can be positive or it can be negative. That if that is positive, it means that DNA is supercoiled positively, but if that is negative, it means that linking difference will show that that DNA is negatively supercoiled. That you can see that this DNA is relaxed, and linking number, it means LK0 is equal to 200. But in this DNA, which is negatively supercoiled, the linking number is 198. That it will produce the linking difference by negative value, so it is negatively supercoiled. But in this DNA, the linking number is 202. That if we minus 200 minus 202 occurs, it means that that if we minus them 202 minus 200, so it will produce a positive value, so it is positively supercoiled. So this linking difference can help us to know that the DNA is supercoiled positively or negatively by this formula, that the linking number in a strand which is supercoiled minus the linking number in that DNA which was in relaxed form. So it can help us to know about type of supercoiling. If you have any question, you can ask and comment. Thank you for your attention and time. Thank you.